Welcome. You're listening to Under the Surface, a podcast where we have in-depth discussions on computer-aided techniques in drug discovery. Welcome, everybody, to this episode of Under the Surface. I'm your host, Chris Williams. I'm joined with a co-host today, Alain Deschênes, not my regular co-host, Dave Thompson, but Alain is gracing us.
Nice to be here, Chris.
Alain is the Vice President of Chemical Computing Group.
Or President of Vice, essentially.
President of Vice at Chemical Computing Group. And we're here today with our guest, Rebecca Swett, who's joining us here in Montreal on one of her visits to Montreal at her new exciting job at X-Chem.
Yes. Thank you for having me.
Thank you. So, we'll start off by telling us a little about yourself, Rebecca. You're a comp chemist. How'd you get started in this?
Sure. How did I get started as a comp chemist? Well, I've been a comp chemist for a very long time because when I was an undergrad, you get to your senior year, you have to pick your capstone project, right? I basically just picked my favorite professor, who is my quantum mechanics professor, because I loved quantum mechanics. And he was like, I'm a physical chemist, so you can either go into the lab and boil naphthalene and take measurements, or we can raid the computer science department, who just got new computers, take all their old computers, make a Beowulf cluster, and we'll do some docking and some MD. I'm like, that sounds like way more fun than boiling naphthalene. So I did that. And from there on in, I was really into the comp stuff. So I stuck around after I graduated because I didn't know what I wanted to do with my life because I was, like an infant. And I taught some labs to just make money because I wanted to take a year off, right? And my department said, you're taking classes. You're teaching classes, so you get classes for free. I'm like, okay, well, I'll take a couple classes. And they said, you get paid more if you're a student and not just an adjunct. I'm like, oh, okay. And that's how I accidentally got into grad school.
Because it had better pay than being a professor?
Exactly. And yeah, so I worked on a master's. I was kind of waffling. And then 2008 hit and I decided, they'll pay me to do a PhD. So I went and started a PhD at Wayne State. And I picked a couple professors that I really liked. And I did the whole usual, you do a little tour of the labs and you sample and try things out. And turns out I am really bad in a wet lab. So after about 3 months of pain with my PI, who is very patient, I just took 2 weeks and I just did all kinds of computational stuff for him. I'm like, this is what I can do. And he's like, do this, stay out of the lab. I'm like, okay, great.
And you were out of the lab because they were doing repair work.
That's an even weirder story. But essentially, I had been doing homebrewing, making homebrewed wine and beer for a while. And they were trying to get me to do tissue culture. And they kept getting all of these weird contaminants. And he went to a conference and came back and he's like, Rebecca, you need to come to my office right now. And I sit down and he's looking at me with this screw face and goes, do you brew beer? And I go, Yes, I brew beer on the weekends. He's like, okay, yeah, you can't do cell culture in the lab because all you're doing is culturing yeast. I'm like, well, I'm good at that.
That sounds like a Michigan thing to do.
It's a very Michigan thing to do. Yeah. And after that, I managed to land the presidential postdoc at Novartis because my other PI, I ended up with two really wonderful PIs, was very awkward. And we were just chatting with one of his old lab mates after an ACS, and this short older man who you might know from Novartis came wandering up, and
would that be Dan McKay?
That would be Dan McKay.
He called him an older man. He's my age. Okay, I'm an older man.
But he wandered up, and I honestly, I feel bad. I can't remember Andreas's former lab mate's name, but she said, he's looking for a postdoc. And Andreas, my PI, looks at him and goes, oh yeah, we have some options open if you're looking, she's like, no, he's looking to hire a postdoc. Andreas turns bright red, shuts his mouth and points at me. And goes and then Dan McKay grilled me for the next 15 minutes relentlessly on MD methods. And he's like, I'm going to come to your poster tomorrow. I'm like, okay.
Yeah, it sounded like a threat.
Yep. And then I ended up at Novartis. After that, I made my way over to Vertex, which was delightful. It was a really great place to learn. And from there, I followed the chain migration from Vertex to Relay, like many, 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 many people did. At one point, Relay was 33% Vertex.
Wow.
And since then, I have made the move over to X-Chem, where I am now, and I'm having a blast.
Well, that was recent. The X-Chem was just
yes. I'm a month and a day. Month and a day.
Yes. But if we go back a bit and talk a bit about Vertex and Novartis, so at Novartis, what did you work on there? Because it was a postdoc, so there was obviously clear goals, right?
Yes. Dan McKay was not prepared for me. He had a project that he wanted to be done about understanding the statistics of water movement. And I love automating things. So I did the entire 1.5 year long plan in 3 months. And then they had to start finding things for me to do because I just automated the bejesus out of everything, made a bunch of nice analysis scripts. And they started putting me on some project work to get me exposure to the different things, which is great. I ended up, because there was actually one of the joint postdocs. So I was technically also with Vijay Pandey for that. Oh, okay. And so we were doing some Markov state modeling applied to some targets that we were looking at. It was just a lot of heavy MD. I was benchmarking a lot of methods. I was writing up a lot of automated scripts for them to pass out to the group who may not have specialized in MD because there's a few in there. But just making things easy and automated and overnightable.
Yeah. So from startup to setup to analysis, running. Yep. Yeah.
It's funny how there's a lot of value in just getting the MDs to run and analyzing them, and it's not trivial.
Yeah. Yes. And I think MD analysis is something that I am very passionate about because if you do it right, you can get so much out of it. And if you just do the bog standard things that they just fall out of the data analysis packages, you miss stuff, So I'm a big fan of fit for purpose coding to answer the deep questions when you have a real mechanistic question. That's one of the few ways to get at it.
And what a more specific example of that? People will look trajectories, look at energies, and look at RMSDs, but I guess you're talking to me about a bespoke angle between this helix and that helix that is probably unique to that system.
Kind of, yeah. There's definitely. I've written several for several different purposes. And I say the one at Novartis that I wrote was basically they had a known allosteric site that was being modulated, that they were trying to modulate, and they couldn't tell from design if they were going to make an activator or an inhibitor or something else. And it's because the RMSDs of the crystal structures, because we had crystal structures of the inhibitor bound, activator bound, RMSD was less than 1.3. How are you going to design around that? It's just all of the data was in the motion, really. Yeah. So just ran a bunch of simulations and and this is very amusing, ended up finding a statistical method from population ecology because I needed to be able to compare matrices of correlations within the simulations. And what do population ecologists do? They go out in the woods and they block off a 10-foot by 10-foot square and they count things in the woods and then they compare the matrices. So there's some really solid statistical methods out there to do this thing that they obviously did not intend it for, but all of the basic statistical assumptions were met with the way the simulations were run, and it worked a charm. And I was able to basically build a predictive model that said, based on your simulation, we're going to collapse all this data. I'm going to match it to the fingerprints of the rest of the motions, and I can tell you what it's going to do. And it works great. And then we automated it and ran it overnight.
Nice.
What year was this?
Oh God, how old am I? That would have been 2013, 2014, somewhere in there.
Back then, were you limited by hardware or was it?
No. And this is why Mark Mirko nicknamed me GPU girl. They put me in and immediately gave me a ton of GPUs just because they wanted to do this. I was very unlimited in compute space, which spoiled the crap out of me. And I was also very unlimited in storage space, which also spoiled the crap out of me. So no, I had a good set of GPUs just on demand. And then when I followed to Vertex, they did the same thing. They're just like, you need MDs, you need to do MD, so you need GPUs, tell us how many. And then they just bought a bunch. So
I guess the advantages of working for big companies, right?
Yes.
And so you said you went to Vertex after this.
I did, yes.
Were you planning on going to Vertex, or it just sort of fell in your lap? Or?
A very good friend used her network to keep me from being one of the many computational scientists who went into data science for finance. I had an offer with an investment company that were going to pay me a lot of money to do data science for investing. And some of it was really cool. And some of it, and actually one of the other postdocs that I work with, Jeremy Beck, did go there. It's the investment firm Transamerica, right? And so he does all kinds of, he was doing crazy stuff with natural language processing to figure out what investment brokers should get what clients. And they wanted a second Jeremy. They wanted a me. And a friend reached out to Georgia McGoy, who she knew would be putting in a position soon. She's like, we're going to lose another one to finance.
Oh, okay.
if you got a position to post, you got to do it. And then they did the sneaky. And my friend's like, you got to go to this BAGIM talk. There's going to be a really good seminar. And Georgia's like, I'm going to post that job as soon as she walks in the door. And she did. And so we chatted, and like a week and a half later, I had interviewed and accepted, and it was everything that I wanted. And it was a great place to learn for a very long time.
So I guess in this field is, when you're in chemistry, you lose a lot of people to med school. But when you make it through chemistry, or comp chem, then you might end up in finance.
Yeah.
I've heard that story quite often, actually.
Yeah, actually, in AI and other computer fields too.
Yep.
Oh yeah, I know a couple QM people from the Schlegel lab who ended up on Wall Street as quants.
Yeah.
You can do rapid, high throughput, high level math. It's a good place to be if you need a lot of money.
Well, that's actually a good theme of this podcast in some ways has been, how do you get into computational chemistry? What can you do with it? And for those of you thinking to study it, well, you can study it. And if you ever get sick of it, well, and you want to make some real money, you can go
you can swap out. No, I mean, I love it. I think I would have really regretted leaving the field just because I've always wanted to do something that helps people, and being in computational chemistry. I'm not the kind of doctor who can like give someone stitches or give them no, gross. Good for them, but no. But yeah, I can make drugs and I can help patients do better. So.
And at Vertex, you were embedded in projects. This was like a regular gig.
Tossed me in the deep end of the pool. I immediately was like toured around to all the different projects. They hadn't hired a new modeler in several years just because Vertex was having a struggle bus with the finances there. They did eventually turn the corner of profitability like 8 months into my tenure. And then now everything is exploded.
Then you got all your GPUs.
They knew I needed those. You're not going to make the good use out of your first molecular dynamics hire without giving me the resources to compute. They were pretty computationally intensive anyway, so it wasn't.
Had that pretty
they did.
Much
They built it in
philosophy, right?
Yeah,
they pioneered a lot of that.
They're one of the first companies to really say that was part of their platform.
Yep.
Yeah. So from Vertex, which is a pretty big biotech, you ended up sort of being attracted by a small company, Relay.
I did, and this is. It was not necessarily the company, it was the people. So Pat Walters, who everybody knows, he is the one who...
Formerly at Vertex.
He hired me into Vertex, and then he hired me again into Relay. He knew that he wanted me on that team, and like I love the sort of organizations that Pat built. He really focuses on like everything is you're embedded, you're supported. He wants you like just hire smart people and let them do stuff, and good things will happen.
And champion science.
Yes, exactly. Just do good science and go out and do things.
And the same thing there, you were embedded in projects working with chemists and all that, that jazz.
Yep, the usual, all the sort of like, if there was a really difficult problem, they'd sort of like lob me at it. But towards the end of my time, not even the end of my time, it was like a year and a half, 2 years-ish. They started bringing up platform initiatives for machine learning, high throughput synthesis, and then eventually the Dell group came up, sort of like at different points. And I am not the sort of person who can see a challenge and not try to fix it. And so I sort of got embedded in all of these things because like there was like the high throughput experimentation group needed a chem informatician, and they didn't have one. I asked one of the guys who was like, he was so excited. He's like, oh man, we got that first high throughput synthesis, 96 well plate. Everything went, it was great. I'm like, great. How'd you pick the reagents? He goes, it was kind of random. I go, absolutely not. And so we talked the next day, and it was very good nature. It was just like this is all the stuff that I can do. This is how much information you can really bring out of these plates. And yeah, I got embedded in a lot of the platform things, and I was really enjoying it. And then when X-Chem came along, it was very like all of that, but bigger.
Well, so X-Chem is known as a Dell company.
Yeah. Yes. So yeah, when you first said Dells, I thought,
what's a Dell?
Yeah, I thought I've got a ThinkPad. What's so impressive about a Dell?
It's funny you say that because I have a Windows machine for the first time in like 18 years, and it's a Dell.
Yeah. It's on brand.
Yes. Yeah, it is on brand. Anyway, a Dell is a DNA-encoded library. And what this means is that basically, if you want to survey extremely large chemical space, we're talking on the order of like tens to hundreds of billions of compounds. What you can do is, it's actually quite clever. Make sure your chemistry is all water friendly. Everything is water soluble. Everything behaves really well. Then you take your building blocks. If you think about clicking together a molecule, like putting together Legos, you have multiple pieces. You can snap them together in any number of ways. And And the way that you get to these really large numbers, and I'll get to why there's DNA in a second, is that if you think about putting 300 piece A in, and then 800 piece B, and then 5,000 piece C,
it's combinatorial
and it explodes. The reason why there's DNA involved is that each of those building blocks is tagged with a piece of DNA. So as you click two building blocks together, you click their DNA tags together, these are tethered with a really long chain. So that's like the compound is sort of floating on this piece of DNA that's in solution.
So it doesn't affect the conformation so much.
It can, so like basically the compound that's on the DNA is pretty far away from the DNA. And you can get interference from the DNA just because it is a huge structure, but that's how you identify like what DNA corresponds to what building block. And that way you can read out the DNA tag, build out the building blocks, and then you know what your molecule was.
So is it, as opposed to building, let's say, bespoke molecules, you just build them all?
Yeah, kind of. That's.
And then when you do your assay, you've got the molecules that bind that sort of still attach their DNA tags. Yep. You pull them out, and you decode the tag, figure out what the molecule was.
You do a lot of high throughput sequencing to see like what all of the compounds that attach to that thing were. And usually when you're doing like an assay like this, you set up multiple different assay conditions. So you have like counter screens and things like that. There's like the first screen that you run is just the resin that you use to pull down. So if you thing binds to that, well, you have no way of knowing if it's the compound or not. So no, gone, bye.
Oh, so you're getting rid of false positives that way by doing these counter screens and orthogonal screens to get rid of noise signals.
To get down to what is actually doing the business. Yeah.
The difference, let's say, in your previous life, I'm sure you did a lot of, structure-based drug design.
Yeah.
To switching to this, it must have been a mindset.
Oh, you think I'm not doing structure-based design?
Tell me.
You're making assumptions, Mr. Vice President.
Yeah, I mean, so right now X-Chem hasn't had computation in a very long time. They had a chem information a while ago. They tried some comp chem, and also X-Chem is a lot older than Relay, right? They've been around for a while, but yeah, so they haven't really had computation, but they've had like all of this data and the explosion of the data over all these years, right? And so just a few months ago, they put in some leadership in a new tech officer, new CEO, new business officer. And these are people that are also from Schrödinger, a couple of them. And so they know computational chemistry, and they look at that data and they go, oh my God, there is so much opportunity. There is so much we can do. There is so much more that you can do with like this size and scale of data with a computer that you can't do just as a human. A lot of what was being done was very manual by very good scientists, by the way. But when you get to the hands of like being able to, how does any one human brain deal with 30 billion compounds? You don't. Okay. And even when you run your screen and you get your hits back out, say you have like a moderately productive screen and you get 80,000 compounds. The human brain, cannot deal with holding that all in your head. And so they have some computational cheminformatics methods in there to work with the data, get their clients what they want. But someone with a computational mind sees it and thinks, oh my God, there's so much more. And the ability to do that large scale with multiple companies that X-Chem is working with, to me, that's a part of what the appeal was. It was that so much broader impact. Like not to be egotistical, I got pretty good at making drugs, okay? But there's only one me. And if I'm going to do this and have broader impact and really help more and more people, I need to go to a larger space than that. And that was the opportunity. And to get all the way back around to your structure-based drug design, that's why I'm in Montreal. Oh. This is why I'm here. A while
structure-based drug design in Montreal?
Could fool me.
Yes. A while back, X-Chem acquired Intellicin, which is a wonderful group of incredibly talented medicinal chemists. And part of what X-Chem is doing is they're wanting to pivot from like this profile of we're going to do some data package, we're going to do some analysis, and here's your hits, sir, to we have really good med chemists who can take these hits and take them to the next level and get to the stage of like, what my personal like sort of brain about this is, I want to get to the second pass compounds first. Because when you get a hit from a Dell, and this is, I don't care who does your Dell, I don't care if it's X-Chem, I don't care if it's Relay, I don't care if it's Novartis, anybody that has a Dell, the compounds that come out, they tend to be optimized for library chemistry, which is to say that the synthesis is prioritized more than drug likeness in a lot of ways. But that doesn't necessarily mean that you're going to have a good starting point. Have you guys ever like had compounds like come out of a DEL that you had to like look at and deal with?
Never.
No, never.
We've had HTS hits back in the old days. Used to be called CombiChem.
Yeah.
Right. And there'd be multiple wells and you'd have to resolve the wells and things like that. And the same thing, you'd have like, 4 positions of functionality, typically with amide chemistry or ester chemistry, quickly, go 4R groups, 1,000 positions each.
So back then it was solid state chemistry, scan the beads, right?
Oh yeah, that's true. That's another thing. But the idea was making these just huge libraries based on combinatorial explosions. But even back then, they found that the compounds that we made were synthetically accessible, but their chemical property space was actually a little bit different from drug property space.
Exactly. And so with all of the information that is in these databases and having this wonderful team of medicinal chemists, I would like to get to the point where we can have second pass compounds first, where you think about even if I wasn't working for X-Chem, this is what I would hope for any DEL, is that you have this rich data environment where you know so much about your screening conditions, your compounds, everything, like looking at what's in there and then looking at where you need to get to. And with computation, trying to get to the clinic faster, because you can answer a lot of questions. Like, for example, if I pulled a DEL hit, right? And we look at it and we assay it and it validates and it's good. You've synthesized it off DNA. It works against your target. And you look at it and go, oh my God, I have a really bad tox problem. This is going to be really problematic. What else could I have picked in that DEL that was not going to have that problem, or what's going to result of, or say you run into a problem that you didn't even know that you had. All of a sudden you're looking at your compounds going, man, I really wish I knew to include this kinase for a counter selectivity piece of that, but you didn't. And now I get to go back and look through what are the alternatives? What else could be in there? What could be helpful? That's just one part of it. But yes, so the structure-based drug discovery in Montreal is very much fueling active projects at the moment. It's pretty awesome.
But it sounds like, my experience, let's say listening to talks from Novartis, is that there's a lot of physics-based stuff that's going on there. And the transition now is that it's a lot of data science stuff. And we are, I think, at the times of data science right now, right? AI, ML. They're not, the two techniques are not at odds, right? They're complementary in a sense. But do you agree that, let's say, what you're doing right now is much more data science, machine learning?
Again, I'd say it's complementary. There's definitely facets of it where the data is so rich. I mean, how could you not resist the temptation to make a machine learning model when I have 80,000 data points, right?
But to use, let's say, physics-based techniques would take a lot of computation power.
Exactly. Yeah. Exactly. And so that's sort of where you have to bring in good judgment and decision making. To me, is when I'm a big fan of, if it works, that's what I'm going to use.
What I think I like about all of this is that it really sort of caters towards experiment, right? Because you make the molecules and you do assays, so you get real data back.
Yes.
So it's not all just predictive data. What you can do with the DELs is, like you said, you have a molecule, let's say a lead, and you can go and see what else is in the pool that may not have the undesirable properties that you're facing.
Exactly. And actually, one of the great applications from ML with all of this data is the idea that and this is a thing that they have, it's called a hit miner, where if you have, say, a certain assay or whatever, and you have enough data and you have your, all of your standards are met, you have your, domain of applicability, you have your 3 logs worth of dynamic range in your data, you make a beautiful machine learning model. And I look back in the database and go there's really not that much there. That was a really weird one-off building block, but it's doing a thing, right? Why not use the machine learning model on Enamine Real and just say hey, $127 a pop, you can buy 50 of these next week and answer your questions.
Yeah. That's definitely more information based, quick, to the result very quickly.
Exactly.
As opposed to, running a dynamic simulation on one compound in a membrane and water and all that stuff.
I am very practical, and that was sort of hammered into me at Vertex with the really, they had an army of medicinal chemists, and it was yes, this calculation is cool. Is it going to make a drug next week? If not, why are you doing it? And that was sort of balanced with the big, longer learnings of okay, this is not going to make a drug this week, but if we really understand this and you give me 3 months to work out what is going on in this system, we will make a drug by the end of the year.
I guess in a sense,
that's where you think of like the data versus like the really big, intense simulation type things.
Yeah. And you're much closer to biology here than you are, let's say, with predictive, let's say, physics-based methods where it's hard to simulate biology in a sense, right?
You need to make good choices for sure. And there's definitely like systems where I don't care how much coarse screening you do, you cannot, I'm sorry, like endocytosis. Clathrin mediated. No, never, ever. Not right now. I won't say never because not right now. Maybe we'll get, our.
Well, it's just a number of atoms time scale.
Exactly.
Right?
Exactly.
We can't simulate the cell atomistically yet.
Right. Guess what I was actually doing in grad school?
Try to simulate the cell atomistically.
That's a lot of GPUs.
That's a lot of GPUs. Yeah, so I think making good choices about your systems, like when you have like large multi-domain complexes and you're interacting with a membrane, that's probably not a great place for physics-based simulation unless you can like dedicate, like you're going to do a thesis on this, plonk, and just make that.
And if you could isolate from that big system, you think the action is happening in this one little region, and I just want to simulate that one little corner, right?
Well, I'm a big fan of like, if you have a simulation that is not physical, but it is useful, I totally game for that. There was a system that I worked with where I was doing all these simulations and said, to understand the mechanism, we don't know what's happening. Okay. So I run a ton of simulations and we see way off in the distance on the other side of our binding, this signal just keeps coming up. And the crystallographer would see it from time to time in her crystal structures. And I would see it very clearly differentiating between actives and inactives in the simulations. I didn't know what the heck it meant. Do I now? Yes, because we got a cryo-EM structure and I did figure it out. I can't talk about it, but it was very cool to see that like chasing that signal, that was a true signal, even though it wasn't biologically like in my simulation, that was what was doing the thing when you have the broader context of the bigger structure.
And were you able to sell all this to the chemists so they sort of came on the ride with you?
That's the stuff you presented at our UGM, right? Where you basically shed like a site and it would sort of deform and the rotation of the side chains would percolate all through the structure and basically affect the interface at a different PPI.
Yep.
Right? So basically, when these things all flipped, all of a sudden it couldn't interact with protein B, even though you're perturbating the side of the protein.
Exactly. And so that was very much just a case of like, let's chase this signal. This signal seems to, it correlates to our data. It very clearly delineates between actives and inactives. What is it doing? I don't know, but I'm going to use it. And that's what we did.
I mean, that's kind of the dream, those kind of simulations where you run this thing and the protein can actually deform and change. And you see this long range effect, a residue change in the pocket that's easy to see, residue change in the surrounding regions, a little bit harder to see, a mutation that's distant that's affecting activity. That's very hard to predict because it's some sort of percolating effect through the protein. And you're going to have to depend upon lots of crystal structures or
and lots of analysis.
Lots of analysis. Exactly.
Yeah, that was a great case where, like, in order to get to that understanding what that signal was doing, we could track the signal. That did not fall out of like the easy data analysis package. That was a custom written, just like.
And that's actually maybe an example of what we're talking about. It's like when you start understanding how your system's working, I need a descriptor that describes how that thing percolates or changes through the structure, and it's not going to be in your out of the box RMSD,
distance metrics, things
exactly.
like that.
You've got to really understand what's going on, create a descriptor to measure it, and then go measure it through your trajectories and start to look. Which is it's good to bring that stuff up too. The dynamics are good, but it's analyzing the trajectory is as hard, if not harder, than running the dynamics.
Yes,
and you code, right?
Yes, I code with bubblegum and duct tape when I need to, when I'm angry enough that something it doesn't exist out there. Like, fine, I will write my own analysis.
And I think that's important. I guess the tools in a sense are getting so easy to use nowadays that you can sort of squeak your way by without being able to code.
Well, there was a big complaint about that too around COVID, where the spike protein comes out. Everyone's running docking studies against it,
and there was so many bad papers.
Yeah, and they're just reporting docking scores. Here's a bunch of compounds with docking scores.
Trying to save the world, right?
That was the year when everybody became a computational chemist.
Yeah. I mean, it's a skill, to become a comp chemist, you have to know the chemistry, which is really hard to learn. That's not a given. The computer side is actually quite feasible, but I think you become I think, Chris, when I met you, you told me something. You said, I know here we're going to plug Mo, but you said I know how to use Mo, but if I use SVL, I can make Mo sing essentially, right?
Yes.
So whatever platform you use, if you're able to code in it,
yes,
you can actually do all these crazy analyses that you otherwise couldn't do.
Well,
absolutely.
It's because it's a science, not engineering. I mean, we're still discovering things all the time. Different techniques are coming out. We see in this field has seen so many different techniques rise and fall in popularity and then rise back up to their,
Yeah, we had QSAR. When I started in this industry, we had QSAR, and now we have machine learning.
Exactly.
So we're back.
And we had combinatorial libraries. Now we have DELs.
Yep.
Right. So there's been definitely repeated cycles. But I mean, it just underlies that drug discovery is extremely difficult.
Yeah. I guess the next thing we'll have is SGI is going to make a comeback in this industry, and we'll all have SGIs again. Who knows?
That's when I retire. Carrying that little 50-pound computer from site to site. When I got my first laptop, that was like a dream. So actually, I do want to get to the things that you've done in the community, right? So you've been active in the comp community. You organized BAGIM in Boston.
I am one of the co-chairs with Brian Kelly, and then Scott Parker is the president, and Anthony Arbonites is our treasurer. So call outs to all my guys.
So tell us for the people that don't live in Boston, or even people who aren't quite in this field yet, what is BAGIM?
Sure. BAGIM is the Boston Area Group for Informatics and Modeling. It is the largest meetup for computational chemistry, informatics, modelers in the world. I have not found a bigger one, and I have looked.
I would testify to that because I've been to other ones. I've been to the SAGM, and they definitely are smaller by comparison. And that's just because Boston, as we know, is the Hollywood of biotech and drug discovery.
Yeah. So it is a nonprofit group that we basically exist to facilitate sharing in the community, networking, getting people to meet each other. And usually during the school year, just because people have parents and, summers people leave, we host seminars, usually monthly, sometimes more than that, sometimes less. And we facilitate networking events, and we try to help, if you have a job and you need somebody to fill that job, we try to provide a board that you can share with. If anybody has any job postings, they could just send it over to the BAGIM officers, and we'll post it on our little meetup website. It's a lot about giving back and giving the community the opportunity to interact with each other. So during COVID, we were full remote, and we started hosting a ton of talks that were incredibly popular because you could do them at noon, and you can get the both East Coast, West Coast, and Europe all calling in. We haven't done any of those in a little bit, but we probably will if there's, if another spike or if there's a demand. If somebody from, Europe wants to give a talk, I'll do one online. I'll kill my lunch break. That's fine.
Yeah, I think people got a little bit tired of online meetings, and that's why it kind of went down. But it is good on occasion to do something like that.
Yeah, and we are back now. We are back in person. I just hosted one 2 weeks ago, something like that, with the ZIFO guys talking about data governance.
That's what I like about it is that, every time I open my inbox and I get an email from BAGIM, it's always a completely different topic.
Yeah.
I mean, Chris gave a talk recently on psychedelics, on modeling psychedelics.
That was great.
It's on YouTube if you want to Google it.
Yeah.
And
it was fun.
Yeah, it was fun.
But it is definitely fun because it's very informal in some ways.
Yes.
And very open.
Yes.
It's and they're not meant to be technological talks necessarily. They're not meant to plug things. They're
Exactly.
Right. We have a very hard, no, this is not an advertising platform.
Yes. Yeah.
If you want to do cool science, no matter who you are, and you want to come tell us about it, that's great. Don't give me a sales pitch.
Exactly.
Yeah.
And I think you should be as strict as possible with that because it keeps it gives it credibility.
Yeah.
Where otherwise it would just end up being bought by.
We get enough vendors.
Exactly. Especially in the Boston area, Cambridge area, there's going to be vendors up the wazoo.
Yeah. Everybody wants your ear and your wallet.
And we've had some really wild out there talks that have been really fun. We had Alex Wiltsko in from Osmo.ai mapping the cheminformatics of scent. Which was very cool.
I have a story about that.
That was I got to go to the scent lab and smell the different smells. Oh, it was so cool.
So when that paper came out, I just happened to be sitting in Montreal in the Air Canada lounge. And the person next to me just started chatting. What do you do? I work in this and that. He's like, oh, I'm a wine taster. And we started talking about data science and all that stuff. And I literally I sent him an email with the paper from the Osmo crowd to show them that it's possible to try to train models to be able to detect scents from chemicals.
Yeah.
And I never spoke to him again, but he looked fascinated at the moment anyway.
It's so fascinating. I'm so glad I got to go. It was like, it works. And these guys are not comp chemists. These are data guys.
Well, I think it would make sense because as a comp chemist, we want to see how that thing interacts with the receptor.
And there's one structure.
And that's really, really complicated.
Yes. No, but also, I mean, I think they've done a really fantastic, they have since hired comp chemists, which is great. Well, you know a couple of them.
I do.
Maybe you should apply to be an experimentalist there, Chris.
All I do is taste and smell things all day long.
I mean, but it was really cool because they basically made these models, predicted on molecules and then went and bought them and smelled them.
Yeah. Well, that's the thing. We were at a company that did fragrances as well. A lot of times our endpoints, especially in physics-based methods, are going to be like KDs or binding energies.
Yeah, but this is very qualitative.
But these methods are like it smells musky, smells fruity. Like there's no math in that at the endpoint whatsoever. So there's no physics thing to train on. Right, so I find that very challenging too, because these things are receptors interacting with other receptors. They associate. So the full atomistic description is maybe kind of out of our reach, like endocytosis.
Yeah.
But data science and maybe a little bit of modeling can help.
Mm-hmm. Well, and what they did was there's not data for this. So they went out and hired trained humans to smell and classify compounds for them.
Yeah, the company we went to has little booths where people sit in there and then they smell stuff, isolated from the rest of the world.
Yeah.
It's interesting. I mean, if you think about it, molecules like smell molecules are just basically drugs for your nose, right? In the end.
Yeah. Well, they hit GPCRs.
They hit GPCRs. They so in the end, they have the same problems. It's a lot of, I don't know what the crystal structure is. I don't know what it looks like. And I have lots and lots and lots of data. Yeah. Right. And a lot of data is qualitative in the sense of like smells good, smells musky, smells.
And the thing is, unlike enzymatic stuff where you just go block the pocket and you're done. This is like goes as how does it tickle the pocket? Like how does it bind in? Is it an agonist, antagonist, or is it an agonist this way, agonist that way? I think in some ways, very much like with the psychedelic targets, taste and smell is a form of consciousness. It's really, really complicated how that stuff works. So I think, having that challenge, you've got tons of data. We can't get a full atomistic model of how this is going. We kind of know we're going to have to be flying by the seat of our pants.
Yep.
That makes it challenging.
Yep.
Another thing I do want to talk about is your participation in the Women in Science in Comp.
Or Women in Comp.
Women
ACS,
in Comp,
right?
yeah.
Yeah, that's just like a woman, the thing, the ACS, Women in Comp.
How do we get in? I'm just kidding.
Yeah, well... Well, this has been active for what I think close to 10 years now. When you guys wrote some papers on women in comp chemistry.
Yes,
that was
the brief survey of women in comp chem at Vertex.
Yes,
that was all the women on George's team. We all contributed a piece to that paper, which was just sort of like a retrospective on like. She said, whatever is on the forefront of your mind, just short form. Let us know who you are. Let us know what you're thinking about, and. Now I'm struggling to remember what I wrote about. I'm pretty sure I wrote on allyship and othering was really what it was. Yeah, that was what that paper was, which is a topic that I really wish got more airplay because it really does affect your career trajectory as a woman. Like when you're seen as not part of the group, and we're comp chem here. It's a niche subfield of a subfield. Mm-hmm. The women are generally outnumbered. And this is this is not to say that anybody's like a bad person, but just like implicit bias is a bias that is implicit. You don't know that you have it until you are like properly trained to recognize it. And allyship was actually one of the topics that Georgia, when she sent me to the Simmons Women in Leadership training, really resonated with me because as soon as I started talking to my chemists about, backing me up in meetings, making sure I get credit for my contributions, every single one of them jumped at it. Because nobody wants to be that guy. Nobody wants to say oh, I didn't realize I was sexist. Well, you didn't realize it because you're not a bad guy and you don't think of it that way. But when somebody else if I say we should do this molecule in a meeting and somebody else jumps in and says, 10 minutes later, we should do this, saying, oh yeah, that was a great idea from Rebecca, and just echo that back to make sure that the proper attribution, that makes a huge difference, especially for, when this was going on, when I was more junior in the field,
Yeah.
I've actually always wondered because, inventorship in drug discovery is actually quite important, right? Whoever gets the credit for so in these informal group meetings, are these things kind of recorded? Is there a way to know who designed something, who synthesized something?
Yes. A lot of that is recorded, depending on how the company is set up. But most places do have a way of recording that. And some people have, the design of the compound as something that you can tag to a person, or always the compound, whoever synthesized it first, that's in their lab notebook, right? That's hard and fast. That's never going to go away. But the idea attribution, most of what I see is that people generally are very respectful about it. Nobody's really being a jerk. Nobody's trying to, I've not been in the horror academic experiences, we're going to cut you out of our patent. No.
That's my methyl. No.
There is, if you can say, yes, I told you that would work. And I have one of those. There was a series, but that's off topic.
It sounds on topic. But that's the thing. There is, you're going to get performance reviews based on how much you contribute, but drug discovery is kind of a group effort.
Yes.
Everyone's brainstorming together. Someone comes up with an idea. Maybe someone else comes up with the same idea a little bit later and forgot that he had the idea because he heard you previously. I think it must take a group of people who can get beyond their egos a little bit to make that actually kind of function. Right.
And there's definitely it's, were you in the room when the idea was being discussed? Yes, you're on the patent. Were you thinking about it and doing things and working on it? If I'm put on a series, whatever happens to that series, even if the likelihood that there would be a patent on my series that didn't include any of my work is nil. Because it would be in there.
The patent's usually complete with everything. They're going to
exactly.
And they don't want any challenges either, right?
Yes, exactly.
So this explains why everyone's in meetings all day, every day.
Oh, okay. See, here it's different. We write code, and the thing is, if I claim that I wrote the code, I'm the one that has to debug it. So we like to pass on credit to other
basically
people.
we pretend we do nothing, and then we're not responsible.
Exactly.
So, Chris, should we land this plane?
I think we should. Well, thanks, Rebecca, for
yeah.
We'll redo this, but I do have one last question.
Okay, there we go.
I do have one last question for you. If you hadn't become a comp chemist, what would you have done?
Are you asking now me or when I picked my undergraduate capstone me?
In that case, I have two questions.
He's both.
All right. Well, I would say if undergraduate me had tried my capstone project with my favorite professor and absolutely hated it, I know exactly what I would have done because I was supporting myself by teaching voice and piano lessons and working as a director in musical theater. And I was fully self-sufficient. I started teaching the labs for extra spending money. That's how the gateway drug into graduate school. But yeah, I likely would have ended up working in theater, singing songs and making my friends dance. Do you think quantum mechanics is hard? I had to teach 120 kids to sing in harmony.
Oh, ouch. Ouch. Well, actually, kids are pretty good. It's adults trying to sing in harmony. That's hard.
Oh, that fun old times. Yeah, I was actually a double major in chemistry and performance piano.
That's a combination. My father would have asked me so what are you going to do with that exactly?
I started college when I was 16. I had no idea what I was doing. I'm
started college at 16. You're like a savant of some sort.
I'm in my 40s. I still don't know what I'm doing.
That's young for college, isn't it?
Yeah. Most people go to 18.
18. Okay. So you skipped a couple grades.
I did not skip a couple grades. My mother noticed when I was very young that I was talking too damn much for someone who was under a year old.
Sent you off to college. And she finally got some peace.
She pushed to get me into school because she knew that the statistics of if you have a bright child and you don't get them in early, they're more likely to drop out. And so she got me into school. So I graduated a couple years early. Didn't skip a grade, just started with a head start.
So then question number 2.
If the sun throws out an EMP and fries all electronics, and there's no more comp chem forever because the sun has declared it so, I would likely start a bakery. I love to bake as a hobby. I do it all the time for my friends. Now do you want to play the game?
Yes.
How many novelty pans do you think I own?
15.
1.
13.
I win.
Chris gets
That's right. Price is Right rules.
Yeah, I who would have thought that someone with a fiddly detail-oriented job has fiddly detail-oriented hobbies like baking?
Well, so you must have liked analytic chemistry.
I do. I love analytical chemistry.
I hated analytical chemistry, but I was a
chemistry.
good bucket chemist. So I'm more into like stir fries and food where the measurements have these big error bars and it's still okay. Baking was more precise.
Oh no, I love it. I love it. And it's funny that you mentioned that, but yeah, analytical chemistry, I absolutely love it. I think it's fascinating. I love all the machines. I love the details of but I get excited.
This whole discussion now makes complete sense. You joined a company in Boston that has an office in Montreal. You're in Montreal right now. And I think it's fair to say that we are the baking capital of North America.
Oh yeah, easy by far. New York may put its hands up, but no, definitely.
Let's go eat bread.
Okay.
Yes,
let's go eat bread about it.
All right. Thank you very much, Rebecca. That was a great talk. Thank you, Alain, for being my co-host. Dave will be upset that he missed this great conversation, but we'll make it up to him someday.
I have to do it. Rebecca, it has been a delightful conversation.
And on that note, thank you all for listening to Under the Surface. Thanks for listening to this week's episode of Under the Surface. Don't forget to subscribe to our podcast on Apple Podcasts, Spotify, and other platforms so you don't miss an upcoming episode. Enjoying our podcast? Please rate and review us on Apple Podcasts and Spotify. Have topics or speaker suggestions for future episodes? Send an email to podcast@chemcomp.com. That's podcast@chemcomp.com. Until then, keep exploring, keep learning, and keep loving the science. Signing off.